Bismillahirrahmanirrahim. Today, our topic of discussion is protein. The name protein was given by Berzelius due to its primary importance. The formula of protein was given by Mulder. Proteins are also called polypeptides. Why they are called polypeptides? Because they have peptide bonds among the amino acid molecule. So, what is peptide bond? Peptide bond is the bond present between the amino group of one amino group of one amino acid, and the carboxylic group of another amino acid, the carboxylic group of another amino acid. And the removal of water is called the formation of peptide bond. So this reaction is condensation reaction or dehydration reaction, because here the removal of water occur. Characteristics of protein. Protein are the most abundant organic compound containing nitrogen. 45% of body's dry weight is made up of proteins. Proteins content in 100 grams of substance is equal to gram of nitrogen divided by grams of sample multiplied by 6.25 multiplied by 100. Proteins, as we discussed earlier, that they are macromolecule, large compound, so it must be formed or composed of small units, so they are called amino acids. Amino acids are those small units which combine to form proteins. So there are 300 amino acids occur in nature. Only 20 different types of amino acids are involved in protein synthesis in human. The general structure of amino acid is that it have a carbon called alpha carbon. This alpha carbon is bonded with carboxylic acid, also bonded with the amino group, also bonded with hydrogen, and the fourth bond is with R side chain or variable chain. All the amino acid have amino group, carboxylic group, and hydrogen, but the amino acids are different from one another due to this R chain or variable chain. So variation in amino acid is due to R group or variable group. There are five types of amino acids, which are non-polar, aliphatic R group, aromatic R group, polar uncharged group, and positive charged R group or basic amine, and fifth one is negative charged R group or acid amines. Non-polar aliphatic R group are those R group which contain non-aromatic compounds, like the example of amino acid and this group are glycine, alanine, valine, leucine, isoleucine, and proline. Note that the simplest amino acid is glycine. Why they are called the simplest amino acid? Because they have hydrogen instead of long side chain. So R is replaced by hydrogen, then this is called glycine. That's the simplest one. If R is replaced by double ring structure, so they are called tryptophan, which is the most complex amino acid. Second is aromatic R group. Aromatic R group are those R group which contain benzene ring or conjugate bonds. So here is the example of amino acid: phenylalanine, tyrosine, tryptophan. The third one is polar uncharged group. The polar uncharged group contain serine, threonine, cysteine, asparagine, glutamine, methionine. The cysteine and methionine contain sulphur. So they have extra bond called disulfide bridge among them, or called disulfide bond. The fourth one is positive charged R group or basic amine. They are called positive charged R group or basic amine because they have electron donating species, nitrogen and R chain or side chain. So they have the ability to donate the electron to the rest of the group. So they are called positive charged R group. They contain lysine, arginine and histidine. The fifth one is negative charged R group or acid amines. They are called negative charged group because they contain electron withdrawing species which in turn increase acidity. So they are called acidic amines. And like they contain COO minus group which is electron withdrawing species. That's why they are called negative charged R group. The physical and chemical properties of amines. Amines are colorless, crystalline solid, soluble in water, amphoteric in nature. Amphoteric means they react as acid as well as a base. Slightly soluble in alcohol, dissolve with difficulty in methanol, ethanol and propanol. The R group and pH of solvent plays important role in the solubility of amines. Isoelectric point. What is mean by isoelectric point? The isoelectric point is the pH at which an amino acid is electrically neutral. So isoelectric point is the pH at which the amino acid is electrically neutral. Here is 3 diagram in which the first diagram shows that here is positive charge in nitrogen. Here is both the positive and negative charge, so it got balance. It is electrically neutral. And the third one shows that it's negative charge anion. The positive charge cation, the isoelectric point electrically neutral. So this is called Zwitter ion. What is Zwitter ion? Electrically neutral amino acid is called Zwitter ion, which have positive as well as negative charge. And the third one is negative anion. So the pH of positive cation will always be less than the pH of isoelectric point, and the pH of isoelectric point will always be less than the pH of anion. Let us give the example of glycine. Here, if we replace the R by H, then it will act as glycine. So the pH of cation of glycine is 3.0. The pH of isoelectric point of glycine is 6.0 and the pH of a 9 glycine is 9. So 3 is less than 6 and 6 is less than 9. Second property is ultraviolet absorption. So the compound having conjugated double bonds have the ability to absorb the ultraviolet lights or ultraviolet rays. The example is tryptophan and tyrosine. They are both aromatic R group and they have alternate single and double bond. Due to this stabilized bond, they absorb the light in ultraviolet region. The third one is ninhydrin reaction. Ninhydrin reaction is a quantitative method in which we measure the chemical test of ammonia, primary and secondary amines and amino acids, which is a redox reaction. And when ninhydrin react with amino acid, the maximum absorption peak is 570 nanometer, and the absorption peak is directly proportional to the content of amino acids. Larger the content of amino acid, greater will be the absorption peak. Deep blue color is obtained called Raymond's purple, and it gives the purple solution. Except all the amino acid gives purple solution except proline and hydroproline. When proline and hydroproline react with ninhydrin, it produces yellow color. Variation in structure and function of protein. Proteins are structurally and functionally different from one another due to the types of amino acid they contain, the number of amino acid they contain, the sequence of amino acid that have, and the number of polypeptides chains. For example, insulin is a protein having 51 amino acid, is different from hemoglobin which have 574 amino acid. Types of peptide chain. We are familiar that what is peptide chain. Now we are going to discuss dipeptide chain. A chain having 3 amino acid and 2 peptide bonds are called dipeptide chain. And a polypeptide chain have many amino acid and many peptide bonds is called polypeptide. Note that the myoglobin is composed of 1 polypeptide chain, the insulin is composed of 2 polypeptide chain, and the hemoglobin is composed of 4 polypeptide chains. Now, the specificity of number and sequence of amino acid and protein is very, very necessary for the normal function of protein. Like for example, it is a blood disorder called sickle cell anemia, which occur due to when glutamic acid is replaced by valine. So the hemoglobin becomes sickle shape and it got the disorder called sickle cell anemia. Now come on to the next topic, monomer protein or monomeric protein. Monomer proteins are proteins having 1 polypeptide chain. For example, lysozymes and myoglobin have 1 polypeptide chains. So peptide chain, only one polypeptide chain. So they are called monomeric proteins. Multimeric proteins have many polypeptide chains. For example, hemoglobin. First class protein or complete protein have all the essential and non-essential amino acids like animal protein, while second class protein or incomplete protein are those protein which have a lack of some amino acid, for example plant protein. We have discussed the kinds of protein on the base of polypeptide chains. So now on the base of structure, proteins are of four types: the primary structure, the secondary structure protein, tertiary structure protein, and quaternary structure proteins. So the primary protein have linear arrangement of amino acid, like one amino acid and two amino acid. Between one amino acid 1 and amino acid 2, there is a peptide bond between the carboxylic group and amine group. So that's why the structural representation is N-terminal, mean here is amino group. N-terminal is amino group and C-terminal is carboxylic group. From amino group to carboxylic terminal, this is the structural representation of primary structure proteins. The primary structure protein have only peptide bonds and their amino acids. Between the amino acid there is only polypeptide bond. There is only peptide bond. Normally primary structure proteins are functionless or non-functional, except insulin, which have hormonal function. Second is, the second type of protein on the base of structure is secondary structure protein. The secondary structure protein in which a peptide chain or amino acid arrange itself in the form of helix or plated form. So they are called, in the form of helix they are called alpha helix, in the form of plated sheet these are called beta plated sheet, or simply beta plated secondary structure. Along with alpha helix and beta plated, secondary structure may also be in the form of beta turns or omega loop, and when many secondary structure, two or more secondary structure combine, they form motif. Now we shall discuss about alpha helix and beta sheet. Alpha helix, the example of alpha helix is keratin, while the example of beta plated is silk fiber. Beta sheet may be parallel or anti-parallel. The R group on alpha helix goes outwards, arrange itself outwards. Here is the alpha helix and there is the R group outside of the helix. So this is the structural representation, and each turns have 3.6 amino acid. In each turn of alpha helix there is 3.6 amino acid, and the distance between two helix or two rings is 0.54 nanometer, and the distance between two adjacent amino acid is 0.15 nanometer. And in alpha helix all the peptide bonds participate in hydrogen bonding except the last and first one. The bond present in secondary structure is hydrogen bond and peptide bond. Like this is the between the amino acid 1 and amino acid 2. Amino acid 1, amino acid 2, there is peptide bond. There is amino acid and there is amino acid. They have peptide bond. And in this loop, this amino acid and this amino acid have extra bond called hydrogen bond, which is a physical bond. The third the third one is tertiary structure protein. When the secondary structure protein organizes itself into three-dimensional structure, they are called tertiary protein. Myoglobins and enzyme are the example of tertiary protein. The bond present between tertiary protein is hydrogen bond, ionic bond, disulfide bond, and peptide bond. Look at this diagram. There are different polypeptide chain which organize itself into three-dimensional structure and have an active site also, so they are specific in function. The fourth type of protein on the base of structure is quaternary structure. The quaternary structure proteins are those proteins when two or more polypeptide chains associated with each other to form large-scale molecules. Example are hemoglobin. The bond present between the quaternary structure is hydrogen bond, ionic bond, disulfide bond, and peptide bonds. For further explanation, if we have a tertiary protein and second tertiary protein, these two tertiary proteins combine to form a dimer, which is a quaternary structure. And when there are three tertiary structure and they form a trimer, which is a quaternary structure. And when there are four quaternary structure combines, they form tetramer quaternary structure. Let us use mnemonics for this structure. We have a snake which plays on the earth. So it's a primary structure. When snake curved its body, it become a secondary structure. When the snake organized its body in three-dimensional structure, it is called tertiary structure. When two snakes combine, they form collectively the quaternary structure. On the base of composition, proteins have three types: simple protein, conjugated protein, and protein derivatives. Simple protein are made up of alpha amino acids, for example, albumin. And albumin, there is subcategory O-albumin, which is present in egg, and lactalbumin, which present in milk, and globulin are the example of simple protein. Conjugated protein, when a protein plus non-protein part combine to form protein, they are called conjugated protein. When protein is attached to non-proteinous part, it is called conjugated protein. For example, here the protein combined with nucleic acid to form nucleoprotein. The protein combined with phosphorus to make the phosphoprotein, and protein mucopolysaccharides form mucin and saliva. And protein mean globin plus Fe iron form are chromoproteins. Protein derivatives have example of protein derivatives are peptones and proteins. On the base of function, we have structural protein which help in the formation of structure like collagen and elastin. We have storage protein like albumin. We have enzymes. We have hormones like insulin. We have immunoglobulin which help in immunity, IGs. clotting protein, fibrinogens, fibrins, prothrombin. Carrier protein are like sodium potassium pump, which involve in carrying uh, a substance from one place to another place and help in transport of gases like hemoglobin and myoglobin. Contractile protein like actin and myosin. Glycoprotein, these are oligosaccharide plus protein which help in cell identifications. MHCs, major histocompatibility complex. On the base of shape, we have fibrous protein and globular proteins. Fibrous proteins, they are linearly arranged in the form of fibers or sheath. They are insoluble in water. They have two types, keratin and collagen. Keratin proteins are found in hairs, nails, furs, claws, muscles, cells, etc. While the collagen protein, they are abundantly present in our body. About 1/3% of body protein are in the form of collagen. It is present in ligaments, tendons, bones, cornea of eye. The globular protein, they are globular in structures. They are soluble in water. Examples are hemoglobin, antibodies, enzyme, etc.